So we finally arrived at Sungai Tua Batu Kia, and this is our water sample location.
This is our mini project. We are conducting jar test. The objective of this jar test is to determine the optimum dosage of coagulant and to analyze data and interpret the results of the water quality parameters from the jar test. For the apparatus part, first we are using water bottle, which is 10,000 ml, 6 1,000 ml beakers, jar tester, WD meter, sample tubes, pH meter, spectrophotometer, aluminum sulfate solution, measuring cylinder, syringe, and cube.
First, we need to label all 6 beakers. Next, we need to wash the inside of beaker by using distilled water.
Then, we pour the water sample into the measured beaker for approximately 800 ml each beaker.
Then transfer it all to labeled beaker.
For this experiment, the coagulant used is aluminum sulfate. Before starting the test, the aluminum sulfate solution was prepared and separated into another beaker containing 40 ml of the coagulant reagent. By using a syringe, the aluminum sulfate coagulant was carefully measured and added into each of the 6 water samples according to the required dosage. The dosage were 0 ml, 2 ml, 4 ml, 6 ml, 8 ml, and 10 ml. The syringe was used to ensure accurate measurement.
Next, we need to turn on the jar test apparatus. Then place all the beakers which are labeled as blank sample and number 1 to 5 onto the machine. After that, drop down the propeller into the beaker. We need to make sure that the propeller are not touching the bottom of beaker. After we place the beaker, we need to press 2 on the machine and then press start. The machine will start a rapid mixing at 140 rpm at 3 minutes.
After 3 minutes, the machine will automatically begin a slow mixing at 70 rpm for 5 minutes. After finish the mixing process, let the water sample rest for 15 minutes.
After that, lift up the propeller to take out the water samples. Carefully remove all the beakers from the jar tester. Don't forget to turn off the machine after use.
To conduct turbidity tests, first we must clean the sample cells using distilled water and label the sample cell. Clean the measuring cylinder and carefully collect 10 ml samples from each beaker. Make sure to avoid disturbing settle flocks. Fill sample cells with 10 ml samples. Clean the surface of sample cell and record reading of each sample cell using a turbidity meter in nephelomatic turbidity unit. For color test, prepare a sample cell with 10 ml distilled water. Set spectrophotometer for program code 120. Insert the distilled water sample into the spectrophotometer to set 0. Finally, insert the label sample cell and record the sample cell color value in platinum cobalt unit. To conduct pH test, we first must clean the pH meter probe using distilled water. Then, we immerse the probe in the sample. Wait until reading stabilizes and produces a sound.
The test experiment was performed to find out the best alum dosage to reduce the turbidity by coagulation and flocculation process. The experiment was conducted to study the effect of different concentration of alum on turbidity, pH, and color. The result helped to determine the best dosage for the best water quality. From the result, a clear relationship between alum dosage and water quality can be made. The increase of alum concentration from 0 mg per liter to 75 mg per liter causes the turbidity to decrease from 4.07 nephelometric turbidity unit to 2.58 nephelometric turbidity unit. This decrease indicates effective removal of the increase to 3.09 NTU at 100 mg per liter and 3.13 NTU at 125 mg per liter, which shows that excess alum decreased the treatment
This
efficiency.
graph shows the color removal at different aluminum sulfate dosages. The untreated sample recorded 61 platinum cobalt. The color would then decrease to 34 platinum cobalt at 75 mg per liter, which was the lowest value obtained. When the dosage increased to 100 mg per liter and 125 mg per liter, the color increased again to 51 platinum cobalt and 53 platinum cobalt. This trend matched the turbidity result and showed that excess aluminum sulfate reduced treatment performance. Based on this result, 75 mg per liter was the optimum dosage for color removal.
This graph shows the effect of alum dosage on the pH of the water sample. The untreated water recorded a pH of 7.57. As the alum dosage increased, the pH gradually decreased to 5.08. This happened because aluminum sulfate undergoes hydrolysis when dissolved in water, producing acidic compound which lowers the pH. Although a higher dosage improved coagulation, it also made the water more acidic. The optimum dosage of 75 mg per liter produced a pH of 5.39.
In conclusion, the experiment successfully identified the optimal alum dosage for water treatment. The best performance was achieved at 75 mg per liter, with the lowest turbidity and the lowest color value. Higher dosage reduced treatment efficiency and lowered the pH because aluminum sulfate becomes acidic when dissolved in water. The treated water achieved Class 2 under the National Water Quality Standard (NWQS) for turbidity and color, although pH adjustment is still required after treatment. Overall, the jar test proved that selecting the correct alum dosage improve water quality, reduce chemical use, and support SDG 6, which is clean water and sanitation, and SDG 2, good health and well-being.